Can I start?
Go.
Okay, thank you. So I'm Wataru Iwasaki from the University of Tokyo, and I would like to introduce our recent study, which is titled Machine Learning Enables Prediction of Metabolic System Evolution in Bacteria, which was led by Naoki, a graduate student in our laboratory, who is a great graduate student. So, du venom nu, ku zom nu, u anon nu. I'm not sure if French pronunciation is okay, probably very bad. But anyway, so where do we come from? This is evolution. And what are we? This is genomics. Then, where are we going? This is what we want to address in this study. That is predicting a future evolution of genomes. So prediction of future evolution has impact in various fields. In evolutionary biology, predicting future is important for revealing what is stochastic and what is deterministic in evolution. And also in medicine, for predicting the emergence of drug resistance bacteria, which is very important in the context of public medicine. This is also a very important goal, which becomes possible by future evolution prediction. And also in bioengineering, predicting evolution is important for assessing whether synthesized genomes, synthesized bacteria are stable or not in the context of long-term cultivation. So we would like to predict future of genome evolution. But to do so, of course, we have to learn from the past to look into the future. So we got a big phylogenetic tree that contained about 3,000 bacterial genomes, and also a high-quality phylogenetic tree from GTDB. And by using the genome data, we created a phylogenetic profile or gene presence absence data that is a map to the KEGG database. And then we reconstructed gene presence absence patterns of each ancestral species by using PastML, which was developed by a collaboration with Olivier Gasquet's group and our group. So we got a phylogenetic profile for each ancestral and extant nodes of this big phylogenetic tree. And then we focused on the branches of this phylogenetic tree. In each phylogenetic tree branch, in the ancestral node or parent node, we have gene presence and absence pattern data. And also we know whether each gene is gained or lost along the evolution on that branch. So we trained the machine learning models, logistic regression and random forest to predict if each gene which is absent in the parent node will be gained in the child node, or whether the gene which is present in the ancestral node will be lost in the child node. So the rationale behind this prediction is that every gene on our genome is not independent. So every gene pair has functional relationship. So absence presence of gene in ancestral node will be useful for predicting whether each gene will be gained or lost in future evolution. So we learned which genes tend to be absent before gene gain or loss of a gene, and we predicted which species will gain or gained or lost a gene from gene set information. And then we did cross-validation by splitting all the branches into test branches and also training branches. And then we trained machine learning by using training dataset and then tested its accuracy. And we quantified it by area under curve or AUC. If AUC goes over 0.5, the prediction is non-random, or the prediction of gene gain and losses were predicted significantly. And we found that for the gene gains, AUC significantly goes over 0.5. That means gene gains were significantly predictable by using machine learning. Gene losses were also significantly predictable, but AUC was lower than that for gene gains. And there may be many questions. So whether it can be questioned that whether a specific functional group is more predictable than other groups. So we splitted our prediction to functional categories and found that the prediction is possible regardless of gene functions overall. But the AUC values were slightly different between gene functions, so that, of course, there are some differences among the gene functional categories. But so evolutionary analysis has many pitfalls or many difficult points. So in case of this type of analysis, it is possible that if a branch from very closely related ancestral nodes are split into test dataset and training dataset, we can predict the future evolution by just referring to the referring from the branch in the training set that is very close to the test date one in the test dataset. So we have to be careful about that there is no overfitting because of evolutionary relationship, because every genome share their evolution history to some extent. To address this problem, we we did hold one phylum out validation where uh, uh, all the branches from one phylum is used for test dataset, and the other branches are used for training dataset. So by doing this, we can avoid the problem from phylogenetic effects. And we found that even if we do this whole one phylum out validation, we still see that gene gains and losses are significantly predictable. And then we proceeded to interpret the nature of gene gain evolution and gene loss evolution predictability. So we had a KEGG database, so we annotated the genes by functional roles. So we investigated if the gene gain prediction and gene loss prediction is affected by, can be interpreted based on gene functions. So a simple hypothesis would be that if there is a gene that has a related role, related function, it's more likely that gene that has a related function will be gained. And vice versa, if a gene which is expected to function together is absent in a parent node, it is expected that the gene will be lost in future. So this is actually confirmed in our dataset. So in the bottom panel, the genes that contributed to the gene gain prediction and gene loss prediction are ordered by the feature importance rank. And the pink colors mean that if whether those genes are in the same KEGG module that share a function, and light blue color means that they don't share their functions. So gene losses are accelerated by the absence of genes in the same function, and gene gains were promoted by the presence of genes in the same module. Because we have KEGG database, which has pathway or reaction information, we further got into the details. So, okay, we know that if there is a gene in the same module is present in ancestral genome, it is more likely that a related gene will be gained in future. But so whether that ancestral gene is in the downstream of the reaction or in the upstream of the reaction. So we quantified whether the ancestral gene, which was useful for prediction of gene gain, was in the upstream of the gaining gene or on the downstream of the gaining gene. And we plotted in the left side, we found that it is significantly clear that the parent gene was on the downstream side of that reaction which will be gained in the future. So the prediction direction is from the downstream to the upstream in the metabolic systems. So this is one example of in a xenobiotic degradation pathway. So the blue colors are upstream reaction, and then the red color is the downstream reaction. And on the bottom side, bottom panel, the presence or gains of the downstream reaction is colored in red, and the presence or gain of the upstream reaction is colored in blue. So there is a clear pattern of red to blue evolution. So this is an example of downstream to upstream reaction. So I guess this is a very reasonable result. So evolutionary constraint by functional dependency is that upstream modules cannot work or contribute to fitness of the organism without downstream modules. So first downstream, then upstream. Here is the order. Actually, this hypothesis was proposed 60 years ago in 1945, the year that World War II ended. And there's a theoretical proposal by Horowitz where those pathways will be evolved from downstream to upstream reaction. So we now confirm this prediction by using the abundant genome data available today. Okay, so we also have many other types of data. So we extended our analysis by combining different types of data. And because evolution is heavily affected by environment, so we first questioned if habitat or environment affected the evolutionary orders or evolutionary prediction. So we first collected metagenomic data that were sequenced or that were obtained by researchers around the world. There are tons of metagenomic data that are in public database, but were forgot after they published papers. So we reused those datasets. So we created a database named Proc Atlas, which stands for Prokaryotic Atlas, 3 years ago by collecting metagenomic data, every metagenomic data available, and classified those data into habitats. That means our Proc Atlas dataset links bacterial genomes or bacterial sequences to habitats, not species. Many typical bacterial databases link sequences to species, but our database, our Proc Atlas database, links sequences to habitats. So now we can map those habitat information to phylogenetic trees so that we can now have phylogenetic profile or gene presence absence information and habitat information for every extant node in our phylogenetic tree. And then we observed if there is a bias in gene gain timings and the estimated changes of habitat. And to make a long story short, we found a significant correlation where a gene gain of specific timing of gains of specific genes corresponded to the habitat change to a specific environment. So in the case shown on the right side, so there was so in this degradation pathway, the downstream gene was obtained in soil environment, and there was a clear trend that the upstream reaction was gained in rhizosphere. So if bacteria move to normal soil to near to plant, maybe some degradation from upstream reactions will become beneficial for their survival. So we also observed the common habitat changes from, in this case, soil to plant for evolutionary pattern to show evolutionary patterns. Also, first we wanted to do future prediction, but of course we don't know the future evolution. But we hypothesized that we still can see ongoing evolution because now we have many genomes from a single species, that is pangenome. So if an evolution is ongoing in one species, it is expected that a part of those pangenome dataset already got that gene. So we questioned if our evodictor or evolution predictor can predict ongoing evolution by analyzing pangenomic dataset. So we collected pangenome dataset and observed if part of those pangenome dataset already have one a specific gene or predict or evodictor can predict that that gene is actually going to be gained. And this was actually the case. So for those ongoing gene gains and gene losses, our evodictor successfully predicted their gene gains and losses. And here the blue, this is a phalangic tree, and the blue birds mean that a part of that species or pangenome have some gaining a specific gene. The gene names are displayed here, and the bar heights represent the prediction gene gain probability estimated by evodictor. So for so for species that evodictor predicted these genes will be gained in the future, actually had that gene in a part of those pangenomes. So our evodictor is available in GitHub. So if you can freely use this tool to do prediction of evolution, so genome evolution for your own dataset. What you need is just phylogenetic tree and phylogenetic profile. So you can apply our evodictor to any dataset of your interest just tomorrow. And also, actually, our evodictor doesn't assume anything about gene gain loss evolution. So this can also be used for phenotype evolution, for example. So if you have phylogenetic tree and some table of phenotypes, that can also be analyzed by this evodictor pipeline, evodictor software. Maybe it can also be applied to culture evolution or somewhere, or other applications. So I'm looking forward to an interesting application by using evodictor. So this was recently published in Science Advances this year. So if you're interested in details, please look into the content of this paper. So these are members from our team. So I would like to acknowledge them and thank you for your attention.
Do we have questions from the audience?
It's just maybe you didn't have the time for explaining more extensively, but in this case, on the pangenome analysis and the prediction of the gains and losses. So I will suspect that there is kind of a minimal number of pangenomes or genomes in the pangenome to be able to actually to have confidence on the expectations, right?
Yeah.
What's that number?
Yeah, we selected 50 species that have at least 15 genomes.
15.
Yeah.
Okay.
This is
and did you
arbitrary
try
number.
with less and more, or maybe if you try with 10, then the AUC goes worse, or what?
Either may be possible. We haven't tried that, but yeah.
Okay. So
we can see
with
that.
15, it works.
Uh-huh.
Okay. Thank you.
Very nice talk. It looks like you have different powerful gains and losses in your next slide. Was I right? And do you have any explanation for why you think that might be the case?
Sorry, could you repeat the question?
It looks like you have some slightly different powerful gains and losses.
Certain parts of, sorry, certain.
So it seems like there might be a difference in the ability to tell whether there's a gain or a loss, or there will be a gain or a loss. Is that the case? And could it be the case, and why?
You mean, so difference between the distributions? No.
Well, so you found it easier. It's easier to predict gains than to predict losses, I think is.
Yeah.
Do you know why that is?
Okay. So because gene gains are more difficult to occur in evolution than gene losses, as you know. So genes can be lost spontaneously, but to get genes, you may get a gene by horizontal gene transfers or gene duplications. So gene gains are assumed to occur only under specific conditions. So we suppose that gene losses are more difficult to be predicted because gene losses can occur without big reason.
So
Thank you.
to add to that, in my lab, we've seen that it's harder to get accurate loss rates. And I think that, Lars, you saw that also, right? Yeah, so that might be an interesting thing to talk about. Do we have a question from Juno? We have time for one more, and then we're going to go drink coffee.
Yeah, I just wanted to ask, you said that, so this is used for predicting gains and losses. You said it could be used for phenotype information. Could this be used to predict essentially SNP mutations? So.
Of course, I think so, yeah.
Okay.
So the microscale evolution can be also analyzed by this software, this framework. It's very interesting. Thank you.